Hello, also welcome to this class on excitation and inhibition within spinal cord. So, this is part 1 of I suppose many parts. In this class we will be talking about inhibition, specifically what is generally called as post synaptic inhibition, or so when I mention just inhibition, we are referring by default to this post synaptic inhibition. So, what does this involve? This involves chemicals or neurotransmitters that function as inhibitory neurotransmitters or perform inhibition on the postsynaptic neuron. So, in general when we say inhibition, we are referring to postsynaptic inhibition, or then a special case of inhibition that could be caused due to excitation. How can this happen? We will see how this can happen in today's class, in this class. And then one example of an inhibitory interneuron performing a critical function. This example is a Renshaw cell. More details on this cell and its function in future classes. So, in general we have seen that an excitatory synapse is one that causes an excitatory postsynaptic potential or an EPSP. What does this involve? Some time ago we have seen, for example, the case of neuromuscular junction. So, whenever acetylcholine is released into the synaptic cleft, the nicotinic cholinergic receptors or the ligand gated ion channels sense the presence of acetylcholine and open and allow a lot of sodium to enter inside the muscle cell, causing an action potential in the muscle cell and then contraction of the muscle, so, excitation, contraction, coupling, etc. So, this postsynaptic potential, if it takes the cell to closer to its threshold, right, this potential when it takes the cell closer to the threshold. So, here is the earlier the difference between the threshold and the membrane potential was so much. Now, because of this the amplitude of the difference between the membrane potential and the threshold is reduced, and the probability that this membrane, this part of the cell or this membrane is going to be taken to threshold and an action potential caused in this part of the membrane is increased when an excitation happens. This is called as an excitatory postsynaptic potential. We have seen how this could be achieved. We will review that in the next slide. So, by the way, let us remember that this is called as depolarizing the membrane, is it not? How is this caused? By an inward current. So, by the influx of sodium, we decided to call the currents across membrane as an inward current and outward current in our earliest classes. The first few classes we said what is an inward current and what is an outward current. Inward current is this current that involves an influx of cations into the cell, or an outflux of anions from the cell. This is called as a inward current. If the opposite happens, right, or if cations leave the cell, when would this happen? For example, when potassium leaks from the cell, potassium channel opens and a lot of potassium leaves from the cell, or when chloride enters the cell. When either of these two happens, an anion enters the cell or a cation leaves the cell. So, an outward current is happening. So, this causes a hyperpolarization, is it not? This is called as an hyperpolarization. This reduces the probability at this point, the probability that this cell is going to be taken to threshold and action potential happening in this part of the membrane is relatively reduced when compared with, say, for example, that part, is it not? Here is the resting membrane potential. This resting membrane potential is taken to a value below the regular resting membrane potential, reducing the probability or increasing the difference between the threshold and the membrane potential. This potential is called as inhibitory postsynaptic potential or IPSP. These synapses that cause an IPSP or a hyperpolarization are called as inhibitory synapses. So, synapses that cause a depolarization or postsynaptic cell neuron or excitable cell, any excitable cell goes to threshold and produces an action potential are called as excitatory synapses, and those synapses that reduce the probability are called as inhibitory synapses. And this inhibition can be caused due to chemicals or can be due to other mechanisms. Okay. So, in this case we have seen an earlier case where acetylcholine acts as the excitatory neurotransmitter, opening sodium channels on the postsynaptic membrane, and sodium serves as the carrier for as the mediator for inward current, increasing the chance that it is going to go to threshold. In the inhibitory case, there may be similar neurotransmitters that cause the IPSP in the postsynaptic cell. Okay, let us see other reasons how this can happen. We saw in previous classes. Just to remind ourselves, I can have several stimulations, several EPSPs one after the other that sum to reach a threshold. If these are arriving from the same spot but are following one after the other temporally, there is temporal summation, or these could arrive at the same time from multiple dendrites, from multiple spaces, from multiple points in space, then the summation happens spatially. We call this as spatial summation in previous classes, is it not? Note here the stimulation, positive stimulation or excitatory stimulation is shown in black like here, and the inhibitory stimulation are shown in red like here. So, every time an excitatory stimulation happens, there is a depolarization taking the membrane closer to threshold, but then whenever an inhibitory stimulation happens or an inhibition happens, it is brought farther away, there is a hyperpolarization. So, this phase that is shown in rose or pink is an IPSP, inhibitory postsynaptic potential, reducing the probability that the membrane is going to be taken to threshold and an action potential is going to be caused in this part of the membrane. Let us say something and we said several of the postsynaptic EPSPs, several EPSPs could combine together in time to cause an action potential, or in space to cause an action potential, or several IPSPs could combine together in time and space to prevent the happening of an action potential. Okay. So, there could be spatial and temporal summation, and that is applicable for both EPSPs and IPSPs equally. So, how does this part function? How does this the inhibitory postsynaptic potential functions? How does inhibition itself function? One simple example is the case of GABA, which is a neurotransmitter or an inhibitory neurotransmitter. This is gamma aminobutyric acid. So, we discussed several cases of several types of neurotransmitters. We said amino acids, we said neuropeptides, right? We gave several examples of each. So, here is gamma aminobutyric acid. That is usually, in the usual case, an inhibitory neurotransmitter. How does it function? We will only discuss an important function here. You see a picture that is depicting the life cycle of GABA, which we are not going to discuss. We will only discuss what it does, because in previous classes we have seen the life cycle of acetylcholine. Here we will only discuss function of GABA alone. What this does is, when GABA sensitive receptors sense the presence of GABA. So, how is GABA itself released? The usual mechanism. So, whenever an action potential arrives, neurotransmitters are packed. Neurotransmitter in this case is GABA. GABA is packed into vesicles, the usual method. GABA is packed into vesicles, an action potential arrives in the presynaptic neuron, and it activates calcium as usual. This presence of calcium is detected by the membrane proteins, and the neurotransmitter is released. However, this neurotransmitter is inhibitory in nature. Previously we talked about acetylcholine. When acetylcholine goes and binds to the nicotinicholinergic receptors or the ligand gated channels, the it causes an influx of sodium. Here we have a chemical which when binds to the receptors, it causes an influx of chloride, or depending on specific cases it could cause an outflux of potassium. Both have, if you analyze, both have similar functions, is it not? An influx of chloride into the postsynaptic cell brings the cell to, makes the cell go more negative, or this is an outward current, is it not? It reduces the probability that this membrane is going to be taken to threshold and an action potential caused in the postsynaptic cell. Same with outflux of potassium. So, there is not going to be sodium that is involved in this case, but rather whenever these channels are activated, potassium either leaves the cell or chloride enters the cell, thus reducing the probability that this cell is going to go to action potential or reach threshold and produce an action potential in the brief time that is concerned. By the way, this changes, this is a function of time. After some time there could be an excitatory neurotransmitter, the receptors for which will also be present on the cell membrane will be detected and will produce, but that in the future. At the particular point in time under discussion, this could cause an outward current, thus reducing the probability that this is going to happen. And how is this receptor functioning? Earlier we said nicotinic cholinergic receptors in the case of acetylcholine. Here there are two methods. One is the ligand gated GABA channel, ligand gated channel, similar to the nicotinic cholinergic receptor, which is also ligand gated channel. The other one involves the metabotropic receptors, or involve what are called as mediators, those that involve G protein. The details I am skipping for brevity. It is sufficient if you know that there is not just a simple method by which this function, there are at least two mechanisms by which GABA could function. The receptors are of at least two types, okay. Those that involve mediators such as a G protein, and those that involve ligand gated ion channels. That note that this ligand gated ion channel is not a sodium channel, but rather can be one of these two, can be either chloride channel that opens inside, or can be a potassium channel that opens on the outside, thus reducing the chance that this cell is going to go to threshold, okay. And note, in many of these cases, in almost all of these cases, this neurotransmitter GABA is inhibitory, most of these cases. So, when I say most, whenever I say most, that means that there are cases where this is not inhibitory. For example, in many insects GABA functions as both excitatory and inhibitory. How is this possible? Also in mammals, for example, there are specific cells, these cells are called as, I think, Chandelier cells, check the spelling, specific cells in which this chemical serves as an excitatory neurotransmitter. So, a particular chemical can function either as an excitatory neurotransmitter or as an inhibitory neurotransmitter. How is this possible? By the way, acetylcholine is in general an excitatory neurotransmitter, but there are cases in which it can also cause inhibition. There are examples of that. We will see that in future classes while we are discussing the specifics. How is this possible? Because the channels that are sensitive to the presence of this chemical, say for example GABA, if there is a channel that is sensitive to the presence of GABA, which opens whenever GABA is there, but allows sodium inside. Let us take such a hypothetical case. If there is a channel which opens and allows sodium to go inside, it is going to cause an excitation. So, it is not just dependent. So, the synapses and their function is not just dependent on the chemical alone, it is also dependent on the particular receptor, on the particular channel that we are talking about. In many cases or in most cases, GABA channels are those that produce an outward flux, or those that cause an inhibition in most cases. So, in general GABA is an inhibitory neurotransmitter, and in general acetylcholine is an excitatory neurotransmitter because it involves the opening of sodium channels inside the cell, so bringing sodium inside the cell. So, in general this is true, but not in every specific case. In biology, exception is the rule. So, for every rule that we speak about, there are exceptions. So, we will talk in, so whenever I say this is a GABAergic neuron, so this is going to cause an inhibition, that is true in most cases. So, by default whenever I am saying this is a GABAergic neuron, you should immediately put a negative sign next to it. You should immediately say that that is an inhibitory neurotransmitter, that is true by default. In specific cases where this function varies, if and when we discuss that specific cases, I will tell you that here is a case where that does not happen, the opposite happens. Please note this exception. Okay. So, in most cases it is sufficient if you know that in most cases GABA is an inhibitory neurotransmitter, acetylcholine is an excitatory neurotransmitter, glutamate is an excitatory neurotransmitter, and so on and so forth. So, this is a case, so what we have seen here is a case in which inhibition is caused due to postsynaptic mechanisms involving chemicals, involving neurotransmitters. And then I said there is a case in which I could excite a neuron to cause inhibition in the postsynaptic neuron. How is this possible? That is the question. So, this mechanism is a very special case. For this, to understand how this functions, we need to know how the release of neurotransmitter itself functions. We said how neurotransmitter release is happening, that is due to the presence of calcium channels on the presynaptic cell, and which release calcium whenever an action potential, whenever depolarization arrives. As soon as depolarization arrives in the presynaptic terminal, calcium is released, and this calcium is detected by the proteins, and chemicals, neurotransmitters are released from the vesicle into the synaptic cleft. This is the mechanism that was discussed. But suppose for whatever reason I am keeping the membrane potential at a sufficiently high value, the arriving depolarization is not going to open many more calcium channels than are already open. So, in other words, suppose the membrane potential say was at that value, earlier the resting membrane potential was at that value, for example, and so suppose this is the membrane potential and I am reaching a new action potential brings the membrane close to that value, right. In this case the difference in the potential is smaller, the number of calcium channels that will open for this difference is smaller, whereas in this case the difference between the potential, the membrane potential and the earlier resting membrane potential is larger. So, the number of calcium channels that will open will be larger. So, the amount of influx of calcium will be larger in this case, which I am going to call as case 2, and this I am going to call as case 1. In case 1 the amount of influx of calcium is going to be smaller, and in case 2 the amount of influx of calcium is going to be larger. And note, depending on the difference in this, difference in the amount of calcium that is detected, the number of the vesicles that are opened up or the amount of neurotransmitter that is released varies. So, in other words, it is not dependent on the absolute value, but rather the difference in the amount of calcium that is detected. Since this difference is going to be smaller in the first case, the amount of neurotransmitter that is going to release is also going to be smaller. So, if I can somehow maintain the membrane at a relatively high value of potential, thus opening the calcium channels, some of the calcium channels already, then the difference that is going to be detected is going to be smaller, effectively preventing many or preventing a lot of neurotransmitter from getting released, or much no neurotransmitter can also, there may be a case when no neurotransmitter will be released because you are already at the maximum. So, that is how is that achieved? By having a neuron that excites this part of the presynaptic terminal and whenever that excitation is coming, these calcium channels are going to open and send in a lot of calcium. But when a new excitation comes from the action potential of the same neuron cell body, this is the presynaptic neuron whose cell body is lying somewhere, but here is a different neuron that is activating this part, right. So when an action potential arrives, it reaches this part or the synaptic end bulb, right. Already this part is excited sufficiently that all the calcium channels are already open. There is not going to be any new calcium channel that is going to open due to the arrival of this action potential. So because of that, no neurotransmitter will be released. Whereas here is a case where there are no such excited state of this part of the synaptic end bulb. So whenever an action potential arrives here, there is going to be more calcium channels that are getting opened, more vesicles that are going to open, and neurotransmitter is going to be released into the cleft, which is going to be detected by the postsynaptic cell. Here is the postsynaptic cell, this is the postsynaptic cell, okay, or these are the postsynaptic cells, that one and that one, these three are the postsynaptic cells. When I am not activating this cell, or when I am not activating this part of the cell, the action potential causes calcium channels to open and neurotransmitter to be released and detected by the presence of these receptors here, and a response from this cell can be expected. So this cell responds, why? Because there is no excitation here that is causing an inhibition, but here there is an excitation that has taken that has practically opened all the calcium channels, everybody is already open, new action potential arrives, no more excitation. New action potential arrives, it is not like I can open any more calcium channels because they are all already open. So no difference in the amount of calcium is going to happen. Because of this reason, no new amount of neurotransmitter is going to be released. So amount of neurotransmitter release is proportional to the difference in the amount of calcium that is present before the excitation arrives and after the excitation arrives. So depending on that, if that difference is going to be small, then no neurotransmitter, or if that difference is 0, no neurotransmitter will be released. So this response or this target cell, this postsynaptic cell will not respond at all. So that means it is possible for me to selectively activate. By the way, I can choose to not activate this. Suppose the cell is present but is not exciting this part, then what will happen? Then what will happen is that what is happening here will happen. So this part of the cell is not excited, so all the calcium channels are closed before the action potential arrives. Then the action potential arrives, calcium channels are opening, neurotransmitter is released. So I could selectively close specific parts, specific neurons using this presynaptic mechanism. So here inhibition is happening due to this presynaptic mechanism, okay, or inhibition is happening due to an excitation. I can excite and ensure that there is an inhibition. So it is a very special case of inhibition which allows us. By the way, this could also have what is not discussed is that it could have another branch, say for example here, that is going to release an inhibitory neurotransmitter like GABA, that causes an inhibition. This is postsynaptic inhibition. This is a chemical that is inhibiting this cell or maybe travels to these cells and inhibiting these cells. That is possible, that is postsynaptic inhibition. This is presynaptic inhibition, okay. So there can be multiple cases of this, okay, or there may be another neuron, better still, there may be another neuron that is an inhibitory neuron, for example, which through which whenever an action potential comes, GABA is released, okay. So now GABA travels to these places and inhibits these neurons and there will be no response, that is postsynaptic inhibition, okay. So fundamental difference, presynaptic inhibition is caused due to excitation at the synaptic end bulb, ensuring that calcium difference remains relatively small and no neurotransmitter or very small amount of neurotransmitter is released, causing an inhibition. Postsynaptic inhibition is that case in which a chemical, a neurotransmitter that is inhibitory in nature is released into the synaptic cleft, preventing the possibility or the probability excitable cell is going to go to threshold and produce an action potential. Okay. So, again the difference is discussed here. So, the difference in the amount of neurotransmitter that is released depends on the difference in the peak to peak amplitude, that is what I was discussing. So, the peak to peak, if the peak to peak amplitude of the presynaptic action potential is relatively small, then the amount of calcium that is released is also relatively small. Okay. So, this varies as a function that is shown here. For example, this is an example data. Okay. So, here we discuss a particular case of an inhibitory interneuron within the spinal cord. So, these inhibitory interneurons are called as Renshaw cells. There are more inhibitory interneurons and other interneurons within the spinal cord, not discussed in this class, but will be discussed in future classes. But just wanted to touch upon one particular example, its functions in reflexes, its functions in other domains to be discussed in future. Just briefly we will touch upon this function. So, Renshaw cells are present in the motor neuronal pole. Where is this present? To remind ourselves, in the, so how are these arriving to the muscles? From the ventral roots, is it not? From the ventral root of the spinal cord. So, these are motor neurons. So, these, whenever a motor neuron is activated, this Renshaw cell is also activated. So, what this does is it activates the Renshaw cell, that is shown as a triangle here. So, that is that one. So, this is activated along with the motor neuron, which is which I am going to circle with blue here for sake of clarity. That is that one. Okay. That is the motor neuron. Zoom this out for clarity. Okay. So, that is the motor neuron that is going to take information to the, and there are two of these. We will consider an alpha motor neuron for the case of, so alpha motor neuron means the one that is going to excite the extrafusal fibers or the force producing fibers, that is going to contract the muscle. Okay. This activates another smaller neuron. Now, I am going to draw that in red. Okay. Which inhibits itself. This is a plus and that is a, in other words, this motor neuron activates this interneuron, which inhibits the motor neuron that activated it. What does this mean? So, whenever this motor neuron is activating the muscle, it activates an interneuron that inhibits the motor neuron that activated it. Why would this happen? You cannot keep increasing the excitation to the muscle forever. You need to control that, because if the excitation level to the muscle goes beyond a particular point, it could cause physical damage, it could cause it could have negative consequences which are undesirable. So, you want to control the amount of excitation that is sent. So, as soon as excitation is sent, an inhibition is sent back to that motor neuronal pool. Actually, what it does is more than this, what is not shown, more than this. So, this also branches out to other motor neurons. So, it actually causes an inhibition. So, that may be gamma, that may be alpha, and other motor neurons of distant but agonistic muscles. So, it is going to cause an inhibition for the action, not just for the cell. So, if I am flexing, for example, if I am performing flexion, Usually I give this example as if there are only 2 muscles that are involved. Actually there are multiple muscles that are involved in this elbow flexion function. There are 3 muscles that are involved: biceps brachii, brachioradialis and brachialis. These are the 3 muscles that are involved. When I am flexing, the command for that flexion is given by a motor neuron, and that information is received by the muscle, but simultaneously it also activates this inhibitory interneuron that is going to inhibit that motor neuronal pool. So, it is going to inhibit the whole pool or a large number of cells in this pool. Many of the cells in the pool are going to be inhibited by this guy. This cell that produces an inhibition, that causes an inhibition in the motor neuronal pool. By the way, whenever we say motor neuronal pool, this could be any motor neuron. This could be alpha motor neuron of that muscle, gamma motor neuron of that muscle, and alpha and gamma motor neurons of the other agonistic muscles. So, agonistic muscles are those that perform similar function. So, whenever biceps is activated, it could, or this inhibitory interneuron could inhibit the function of not just biceps but also brachialis and brachioradialis. So, it sends it inhibits a whole bunch of neurons, motor neurons. So, in a way to perform the important function, the critical function of negative feedback. So, I am interested in reducing or controlling the amount of excitation that is sent to the muscle. So, I am interested in placing a roadblock, or interested, this is like having a speed breaker, or so in some sense I want to ensure that it does not increase to a level where I cannot control this. This kind of inhibition that is caused due to excitation. Why, by the way, who is activating this? To remind ourselves, who is activating this inhibitory interneuron? The same motor neuron. It is this cell that is exciting, is it not? So, the cell that is exciting it is getting inhibited by itself, or in other words, it causes, this is called as recurrent inhibition. Okay. There may be cases where the other interneurons may be there. So, this kind of this interneuron that causes inhibition of the same motor neuronal pool that activated it is called Renshaw cell. This interneuron is called as Renshaw cell, okay, already written elsewhere, also written here. There may be other interneurons not shown in this picture that will inhibit the opposite muscle, antagonistic muscle, or that may excite the opposite muscle. When that happens, right, that is different. That is different, will be discussed in future classes. Here we are discussing one particular case. We will just discuss the case of one level of hierarchy in which negative feedback is introduced. There are multiple levels of this negative feedback that comes into the picture. So, inhibition is present at various levels. This is the lowest level at which it is present. There are other levels. So, we will discuss each of those levels in future classes. So, to ensure why so much inhibition, why so much control, why so much control over how much excitation is going, because it is necessary for us to perform functions, motor functions at that level. Maybe there is some demand, there is a necessity for us to perform motor functions at that level that requires control at multiple levels. So, maybe control or negative feedback at multiple levels functions to ensure that the excitation level does not go beyond bounds, does not go beyond limits, causing physical damage. Note, the muscles is made of materials that could undergo physical damage, or we want to at least minimize the amount of fatigue that is felt, or at least other such things, other such considerations. So, which is why negative feedback is present, that is enforced by this particular cell called Renshaw cell. Its function is more functions of this particular cell will be discussed in future class, or way more complications are going to arrive as we continue the discussion in future classes. Okay, so, what we have seen is a particular case of inhibition that is caused due to chemicals such as GABA that are mediated by the presence of postsynaptic ion channels, postsynaptic channels that open due to, that are sensitive to the presence of this chemical, that are called as postsynaptic inhibition. or inhibition that is caused due to an excitation, that is ensuring that the calcium channels are already open, or the difference, peak to peak difference remains sufficiently small that the amount of neurotransmitter release is sufficiently small. This is called as presynaptic inhibition, that can be selectively opened and closed, selectively switched on and off. So, presenting one more layer of control, one more method by which you could switch on and off specific synapses. And then one particular case of inhibitory interneurons called as Renshaw cells that inhibit the motor neurons that activated it, okay, called as, this is also called as recurrent inhibition. In future classes we will be seeing other cases that are called as 1A interneuron, okay, and that what is its function, and other inhibitory interneurons to be discussed, others, not giving the specifics. We will reserve those discussions for future classes. And the various loops of excitation and inhibition, which particular part of the spinal cord is responsible for what excitation and inhibition, we will discuss in future classes. So, we will stop here with this, come to the end of this class. So, thank you very much.